Right, good afternoon, everyone. My name is Monika Myszczynska, and I'm a postdoc at the University of Sheffield Institute for Translational Neuroscience, or Citron. Thank you for the organizing committee for inviting me to speak to you today about how the lab I'm a part of uses artificial intelligence and machine learning approaches to uncover new targets and therapies for amyotrophic lateral sclerosis, or ALS. So briefly on ALS, since Ken Webb spoke about it at length yesterday, it's a debilitating neurodegenerative disorder causing the death of upper motor neurons in the brain and lower motor neurons in the spinal cord. It's characterized by incredibly fast progression and poor prognosis, with an average age of onset in the 6th or 7th decade of life. The vast majority, or around 90% of all patients, are termed sporadic because they do not have a family history of the disorder, with the remaining 10 presenting as a majority a hexanucleotide repeat expansion in the C9orf72 gene. And currently there are around 30 genetic mutations identified as being linked to ALS, and there is no cure available. So on top of the complex genetics of ALS, ALS is also incredibly heterogeneous and complex on the cellular level in terms of its pathogenesis. But the main take-home message from this slide is that even though it's the motor neurons, so the big green cell on the schematic, die and perish in the disease, other glial cells of the central nervous system have been shown time and time again as active contributors to this motor neuronal death. And in today's talk, I'll be mainly talking about those red cells right here, so the astrocytes. Even though it's a complex disease, what 97% of all ALS patients share is the presence of TDP43 proteinopathy. TDP normally resides in the nucleus and shuttles to the cytoplasm under cellular stress, where it incorporates into stress granules to prevent translation of nothing but the most essential genes. However, in ALS or other lysosomal disorders or chronic stress, TDP gets stuck in the cytoplasm, never to return to the nucleus. So in this IHC image on the right, you can see a complete loss of TDP signal from the nucleus and deposits in the cytoplasm. And once in the cytoplasm, TDP gets hyperphosphorylated and cleaved to produce fragments of 45, 35, and 25 kilodalton, which are shown on this Western blot on the left. And those cleavage products are very sticky and act as a seed for former accumulation and further exacerbation of the cell toxicity. Like I mentioned at the start, there is currently no cure for ALS, and the current treatments are not very effective. Riluzole, so the current golden standard of care, was introduced in the mid-90s, and it wasn't until 2017 that the second treatment called edaravone was introduced onto the market. And in that period of time, in between those two drugs, over 60 molecules have been investigated as possible treatments, and the vast majority of them across multiple clinical trials have failed, which prompted us to ask a question: how can this process be improved? And can we harness what's proverbially now known as the golden age of technology and information? And can we use machine learning and AI to try and get to the bottom of this? And maybe we already have the answer. We are on a weekly basis flooded by new publications, new databases of patient and chemical informations are being produced or being updated. And it's definitely a lot more information than a single scientist or a team of scientists even can stomach and make sense of in their lifetime. So having access to an algorithm which can go through all of this information, make sense of it, and return it to scientists in a far more manageable form would actually be very desirable. So as a part of our collaboration with a company called Benevolent AI, based in London, with an office also in New York City, we have done just this. So here are just some of the examples of publications that already have utilized AI to further our knowledge of neurodegeneration or to find treatments. However, the way we do it is we have a trained algorithm which can read sentences and actually understand what the sentence says and extract relationships between different entities that we're interested in. So example of sentence number 1, SOD1 mutation affects ALS phenotype in human motor neurons. Now the algorithm would recognize the verb affects and try and build an association between SOD1, which is our gene of interest, and ALS, which is our disease of interest. So the algorithm can understand that from this sentence alone, it can try and understand a little bit more about SOD1 and or ALS. So it will be deemed a positive relationship. However, in sentence number 2, a researcher investigating SOD1 is suffering from ALS. That relationship is not obvious. So even though the two entities we are interested in occur in the same sentence, the algorithm would disregard such a sentence as it's inconclusive. So all of this information helps us build something called a knowledge graph, which looks more or less like this. So our starting entity is ALS, but it can be any disease you're interested in. We prompted the algorithm to find us a gene or protein associated with ALS, and also bring into the mix another gene or protein associated with our first one, which does not yet have a link to ALS. We want our different genes of interest to belong to different protein families, but we want them to have associate annotations to the same gene ontology terms. We want one of our gene of interest to be a player in a major signaling pathway that the second gene of interest is not a player in. And for the purposes of basic biology, we could just stop right there if we were after new targets. However, since we're doing a drug discovery project, we wanted a link between our gene of interest and a drug via a known drug mechanism of action. And we also wanted the same drug via experimental interaction to be associated with our second gene of interest. So I hope I managed to convince you that this process, even though done by a machine, is actually entirely human-led and informed by expertise of the user. So it is not the case of a giant computer where you press an enter on and it does everything for you. Your knowledge and expertise is essential, but the set of commands is extremely simple and you can recognize and play around with as many novel connections as you want. So in the study that we are conducting at Citron, the way this has worked for us so far is that it has taken the pre-seed system just 20 minutes to identify over 100 drugs of potential repurposing opportunities for ALS. And after a further refinement of just little over a week, we have narrowed that list down to 5 drugs that we've validated in our in vitro model of ALS. Now, this is unprecedented speed. So all of those drugs have already been used on the market. So we are mostly talking here about drug repurposing rather than drug discovery. And drug repurposing has many benefits over de novo drug discovery, as in it increases the efficacy of the whole process, as the compounds are already in use in humans. So the compounds already carry a proven safety record. And because the process takes much shorter, we are also cutting costs. However, there are a lot of different caveats to consider. We would still need to conduct clinical trials, especially taking into account the different side effects that a drug designed for use in the periphery might have in the central nervous system where it has not previously been studied, if it even manages to get to the central nervous system, as brain penetrance very often is a major pitfall in similar studies. But as a whole, as a lab, we would imagine a future where from a cohort of very heterogeneous patients, we would manage to harvest as many information from them as possible and stratify them so that a group of patients which we would deem the best possible responders would be assigned either a personalized drug regimen or the repurposed compounds based on the genetics and other clinical information. So with that in mind, my project aims were to identify novel drug targets and treatments for ALS, as well as their mechanisms of action. And as a whole, on the grand scheme of things, I wanted to assess if those artificial intelligence-driven approaches could be used as a tool to improve drug discovery and drug repurposing for ALS. I've already mentioned our Citron model of ALS, which is an in vitro model based on direct reprogramming of human patient fibroblasts into induced neural progenitor cells. In this model, we take advantage of an unstable intermediate stage that fibroblasts go through on their way to becoming iPSCs. So those cells are not iPSCs per se, as we do not take them back to the embryonic lineage. We introduce neuralizing conditions at the unstable intermediate stage to help them retain their epigenetics and therefore their aging signatures so that they are as close to what the human body was like at the time of biopsy collection. And once we have the pool of fully reprogrammed iMPCs, we can obtain neurons, astrocytes, and oligodendrocytes in a far shorter window of time than you normally would from iPSCs. So for example, astrocyte differentiation takes just 7 days. So how do we use those cells? We assemble them in a pathophysiologically relevant in vitro co-culture assay, which recapitulates the toxicity of astrocytes against motor neurons. So out of the 7 days of differentiation, on day 5, we lift the astrocytes and plate them into 384 well plates. 24 hours later, we treat them with various drugs using this Echo 550 liquid handler, which administers minute amount of drugs into individual wells. And whilst all of this process is happening, we are also differentiating mouse embryonic stem cells into motor neurons. Those cells carry GFP under the motor neuron specific HB9 promoter. So the more motor neurons we get within the embryoid bodies, you can see in the images at the bottom, the more green they will be under the microscope. At day 7 of both of these processes, we bring those two cell types together and we image those co-cultures for 3 days, once every 24 hours to monitor cell survival. So the final readout for, let's say, a successful candidate drug would be an improvement in motor neuronal survival at day 3 compared to the vehicle control. So the compounds identified by AI that are investigating in this project are Gafitano, which is marketed as Iressa, and it's an EGFR or epidermal growth factor receptor inhibitor used primarily for non-small cell lung cancer. And it has been shown time and time again to be a prolific autophagy activator. The second compound is called Nelotinib, which is marketed as Tasigna for chronic myelogenous leukemia, and it's an inhibitor of the BCR-ABL1 complex kinase. And it has previously and is still being investigated as a potential therapeutic target for Parkinson's disease. When we've put Gafitano through our co-culture pipeline in our patients carrying C9orf72 mutations, as well as in the cohort of our sporadic patients, we have noticed that compared to the untreated and vehicle controls, Gafitano-treated cells have significantly improved the survival of motor neurons at the higher doses in the 5 out of 6 patients tested here. Similarly, with Nelotinib, especially at this one dose showed here, we have also shown a marked improvement in the survival of motor neurons grown on patient cells. Like I said before, ABL1 kinase is a primary target of Nelotinib. However, some of our kinome screening studies have also shown Gafitano to be targeting ABL1. ABL1 has previously in other studies been shown to be upregulated in ALS motor neurons, but astrocytes have not yet been studied for the ABL1 burden. So I've tested a presence of ABL1 in our iAstrocytes from a cohort of sporadic patients, as well as our familial C9orf and SOD1 patients, and have shown a significant increase in the expression of ABL1 compared to controls. To measure the target engagement, I have treated those cells with Gafitano and Nelotinib, followed by measurement of ABL1 expression levels, and I've seen a very differential response of those very same cells to both drugs, with Nelotinib showing a trend towards an increase in ABL1 expression and Gafitano towards a decrease of total ABL1 expression. We are still working on figuring out exactly why that is, but some of our preliminary data would suggest that Nelotinib is working more on dephosphorylation of ABL1, which is its active form. Therefore, we are left with more total ABL1. The rest of the talk will mostly focus on TDP43, like I've introduced at the start. So just a short recap: in a healthy cell, TDP is in the nucleus. However, in ALS, we get loss of TDP from the nucleus. We see its expression in the cytoplasm, where it also gets cleaved, producing those, mostly in our model, TDP35 fragments. So ours is one of the very few models out there of ALS which managed to recapitulate this proteinopathy at baseline without a need for any further stresses. So in the image of the left-hand side, the arrows are pointing towards some of the aggregates of TDP43 in the cytoplasm of patient cells. Whereas if you looked on the right-hand side, in some of our sporadic lines, we also see a loss of TDP from the nucleus. Likewise, we also see production of the cleavage product, which is significant in all of our patient cells compared to controls. And also, interestingly, we see most of our patients to have decreased levels of total TDP43 compared to controls. When we tested if Nelotinib could potentially have any effect on TDP43 proteinopathy, we saw that it managed to decrease levels of TDP35 burden in just 2 out of our 3 sporadic patients, whilst having no effect on C9orf72 lines. However, Gafitano has been shown to be potent at reducing levels of TDP35 across all of our patients tested here, and it's also significantly increased levels of total TDP43 in some of our lines. This led us to hypothesize that maybe if we made the Gafitano treatments more frequent or more prolonged, we would manage to completely obliterate the burden of TDP33 in some of the lines. For that purpose, I've tested one C9orf72 carrier line treated at days 2, 4, and 6 of differentiation, day 4 and day 6, and the standard day 5 48-hour treatment, which mirrors the conditions in the co-culture system. However, from the results, I can conclude that the single 48-hour treatment at day 5 represents the total maximum effect we can obtain, as the more frequent treatments did not have any further effect. We wondered, because of the loss of the TDP35 from the cytoplasm and an increase in TDP43, we wondered if perhaps we managed to restore some of the full-length TDP43 back into the nucleus, that we are not dealing just with clearance of the cleavage products, but TDP43 can regain its nuclear function. So for that purpose, we've performed nuclear cytoplasmic fractionation, where we have observed that Gafitano treatment managed to decrease levels of both full-length TDP43 and TDP35 in the cytoplasm. However, it had no effect in the nucleus. So we are getting rid of those fragments in some other way, other than just relocalization of the full-length protein. I mentioned at the beginning that Gafitano is a potent activator of autophagy in the context of cancer. So for those of you unfamiliar, a quick recap on the autophagy system, which is the cell's method of degrading protein aggregates and faulty organelles. So all of the faulty cargo gets enveloped in this autophagophore, which is lined with LC3-2 receptor, which then binds to P62 receptor that's already bound to the faulty cargo. And those formed autophagophores then lies with lysosomes, and degradation of the cargo occurs within those autolysosomes. For the purposes of the assay which I performed, I will explain the next step to you. So in the assays where you introduced an autophagy inhibitor, such as baphyllomycin, what we see is an accumulation of the autophagophores and therefore upregulation of the LC3-2 levels compared to when autophagy is occurring. So in the presence of both baphyllomycin and an activator of autophagy, we would see more LC3-2 accumulation than in baphyllomycin A1 treatment alone. So when conducted in HEC293 cells at baseline, just to verify its function, we've tested Nelotinib for its autophagy function first as well. And as you can see, when we compare the baphyllomycin and Nelotinib co-treatment condition just to baphyllomycin conditioned alone, we do not see any indication that Nelotinib could be an autophagy inducer. However, Gafitano is a potent activator of autophagy in HEC cells in our hands. Therefore, we've managed to run this assay also in some of our control astrocytes, which replicated quite faithfully the experiments conducted in the HEC cells. However, because of the use of baphyllomycin, we could not fully conclude that autophagy is going to completion if we're not just causing start of the autophagy cascade without fusion with the lysosomes. Therefore, we have conducted some imaging studies where we have quantified the correlation of LC3 signal with LAMP2, so the lysosome marker. And in both of our controls, specifically in some of our C9orf72 expansion carriers, Gafitano treatment leads to an increase in correlation of the signal with LC32 with LAMP2. We tried to see if that increase in autolysosomal fusion correlates with the rate of clearance of TDP43. And indeed, we see that after Gafitano treatment, we've got more TDP43 C-terminal fragments, so those that are the most toxic when in the cytoplasm, co-localize with LAMP2, as you can see in some of those images pointed by arrows. So Gafitano not only increases autophagy, but this is also the process via which it degrades autophagy, it degrades TDP43, which might be why it helps motor neurons survive better. So encouraged by those results, we have taken Gafitano into a proof of concept study in SOD1 G93A mouse model of ALS. However, an important thing to point out here is that Gafitano is a substrate of some of the blood-brain barrier receptors, mainly PGP and BCRP, meaning that it gets pumped out of the CNS as soon as it gets in there. So we have decided to create this co-dose condition of Gafitano with a compound called elacridar, which inhibits BCRP and PGP pumps, therefore consequently increasing the free Gafitano in the brain concentration, allowing it to hit all of its targets. So in this study, Gafitano and elacridar co-dose caused a delay in neurological symptom onset in those mice compared to elacridar treatment alone, which is its own vehicle. And we have decided to look at the overall motor neuron health in the mice across all of the conditions by staining them with SMI31, which is the phosphorylated version of SMI32. And in this condition, we see that SMI31 area in the Gafitano and elacridar conditions is significantly smaller, which would indicate a much healthier remaining motor neuronal population in the spinal cord. So as a summary, both Gafitano and Nelotinib improve motor neuronal survival in our co-culture system, recapitulating ALS pathophysiology. Both of those compounds target ABL1, but they have very differential effects on the ABL1 total protein levels. Gafitano is an autophagy activator, whereas Nelotinib is not. Gafitano produces levels of TDP fragmentation, whereas Nelotinib does so only in a subset of patients. Neither of the compounds seem to be having an effect on the nuclear signal of TDP43, whereas Gafitano produces levels of cytoplasmic TDP, whereas Nelotinib has no effect. So to conclude, both Gafitano and Nelotinib increase survival of motor neurons in co-culture, which could make them very good candidates for repurposing in ALS. And although they share some very common targets and mechanisms, they exert the neuroprotection via completely different mechanisms, which we are now hoping to elucidate in further studies. But as a whole, we conclude that machine learning hypotheses could help us improve the process of drug discovery in ALS by speeding it up and helping us understand better what the modes of actions of some of those drugs are. And with that, I would like to thank you all for your attention. I would like to thank my entire lab, past and present. All of this work was conducted as a part of my PhD, so I would like to thank my supervisors, Doctors Laura Fraiuolo, Guillaume Oberg, Richard Mead, and Peter Richardson, as well as all of our collaborators at Benevolent AI. Thank you, and I'm happy to take any questions.
Thank you so much, Monika, for your wonderful presentation.